Hello students, welcome to LEELA'S tutorial. So today we'll discuss about selection and surface sterilization of explant. So how you define explant? It is a part of plant body or the plant tissue which is used as a starting material for plant tissue culture. Right? So here the inoculation of an explant, right, will be done after you select and surface sterilize it. Okay, let us see how you are going to select the explant. So first one is the factors involved. So based on these factors, you are going to select the plant from which you are going to take the explant. Right. So first one is the physiological age. So here it is not the age of the plant you are going to consider, but you are going to check for the physiological age of the leaf, flower bud and all. Right. So then it is the season in which the plant is obtained, the location and the size. Right. So here size also matters because based on the size of the explant, if the meristem, if you are taking the explant is the meristem, then in some plants the meristem is very much minute, very difficult to handle and also difficult to inoculate. In that case, you are going to take any other part of that plant body like nodes. Right, axillary meristem, like that. You're based on the size also, here you're going to select the explant, which part of the plant you're going to take. Right, so after selecting the plant and which part to be taken, here second one is the part of the explant which is used as explant. So based on this size, location, season and the age of the plant, you're going to select the explant. Right, again explant, it is any part of the plant body. What it can be? It can be a flower bud, a leaf section, root fragments, or it can be a shoot fragments. Right? Otherwise you can take the meristem, nodes, petals. Right? So like this you can take any part of the plant body as explant. But here you are going to select your explant based on what is the purpose of doing your plant tissue culture. If you are going to go for the micropropagation in order to produce more number of plantlets, then you are going to select one part. Otherwise, if you want only the secondary metabolites to be produced, in that case you can take some other protoplast and all. Right, where you go for the suspension cultures for the production of secondary metabolites. Right, so based on the purpose of your plant tissue culture, you are going to select the explant. Right, and also you are going to go for the plant tissue culture, whether it is a suspension culture or a semi-solid culture. Right, so everything must be decided first, then you are going to inoculate in that specific media. Before inoculation, the very most important step is surface sterilization. Right? We have prepared the media, the macro elements, micro elements, auxins, cytokinins, everything. Right? So all the proteins and also sterilized by using your filter sterilization. Right, so now the complete media and the apparatus are in sterilized condition, but here the explant which is exposed to the natural environment, right, to the dust particles, airborne microorganisms or any other contaminants, that must be avoided in order to avoid the contamination of your plant tissue culture. Right, so here for that purpose you are going to surface sterilize your explant. Right, so surface sterilization, first you are going to decide which part of the plant you are going to take. Right, so that explant must be excised to 3 to 4 centimeters. Once you excise that one, you are going to wash them under running tap water. Right, for 30 minutes. So when you place these explant in running tap water for 30 minutes, what happens? All the dust particles which are present will be washed away. Right. Then next is soaking in disinfectant. Okay, and mostly we make use of Chlorix. The Chlorix can be used as disinfectant, where you are going to add 3 drops of detergent. Detergent can be tween 20 or you can make use of sodium dodecyl sulfate. Okay, mix them, Chlorix and tween 20 mixed together. Right, then it is used as used for the soaking of explant. So once you are soaking these, then you are going to again rinse them. Okay, no remnant of these disinfectant or the detergent must be present, right? So for that purpose, you are going to rinse them in sterile distilled water. Again, you are going to take the beakers, the 3 beakers with sterile distilled water, right? The sterile distilled water means you are going to autoclave these distilled water, right? Then you are going to pour them in 3 beakers and transfer the explant from one beaker to the another beaker. So it is nothing but rinsing in sterile distilled water for 3 times. So after rinsing them, you are going to transfer them into another beaker. Right here again the beaker consists of 70% ethanol in it. Okay, simply dip the explant with the help of forceps and then again transfer them to the surface sterilant containing beaker, right? So here the surface sterilant is nothing but surface sterilizing agents. So here you can make use of mercury chloride, right? You can make use of mercury chloride. Otherwise, you make use of silver nitrate, hydrogen peroxide. Okay, you can make use of a hydrogen peroxide, bromine water. So you have so many surface sterilants which can be used. Not all together, any one of the surface sterilant can be selected and you can soak them. Mostly we make use of mercury chloride, right? So here mercury chloride, Hg. So here what happens, once you soak them in the surface sterilant or surface sterilizing agent, all the microorganisms or the contaminants will be killed. Okay, now we don't want any remnants of the surface sterilizing agents to be present. So what we must do, again we must rinse them in sterilized distilled water. So again you take 4 to 6 beakers of sterilized distilled water, then transfer the explant from one beaker to the another beaker. Okay, so now this will be rinsing off the explant so that all the surface sterilant present will be washed away. Okay, then it is air drying. Now your explant is completely sterilized, free from any contaminants. Okay, so now you are going to take a petri plate, sterilized petri plate, and you are going to place a stack of filter papers over it. Okay, then place your explant. It can be a leaf or anything, whatever explant you have surface sterilized, that sterilized explant will be placed in it. So everything will be performed in laminar airflow chamber, right? So once it is air dried, now these explants will be transferred into plant tissue culture media, right? That is nothing but your inoculation of the explant in plant tissue culture media, followed by incubation. Is that clear? If you have any doubts, you can put it in comments and subscribe for further videos.